Assalamu alaikum everyone, this is Dr. Manu Bangash. Welcome to my YouTube channel. Hope you guys are doing well. The topic of this video is ion channels, and I'll also be discussing the gating of ion channels. So let's get started. Okay, channels are transmembrane proteins through which ions and molecules are transported down the concentration gradient without utilization of energy. There are also other types of transmembrane proteins called pumps, and the difference between channeled and pump is explained in the video of active transport, so you guys can check that video. Ion channels are the transmembrane proteins or the integral proteins that span through the cell membrane, allowing the passage of certain ions down the concentration gradient without utilization of energy. Okay, as mentioned in the definition, that ion channels are the transmembrane proteins or integral proteins. This diagram shows lipid bilayer, and these are those integral proteins. Integral proteins are those proteins that are embedded in the cell membrane and they'll protrude all the way through the lipid bilayer. And that these integral proteins, they span through the cell membrane. This kind of arrangement of the proteins in the cell membrane is called spanning. This is spanning through the cell membrane. Properties of ion channels include ion specificity, which means that these ion channels are highly selective for the passage of ions or molecules. For example, sodium channel will only allow the passage of sodium ions. Potassium or chloride ions cannot pass through the channel of sodium. Second property is downhill, which means that the passage of ions or molecules through the channels are downhill from high concentration towards the low concentration. And there is passive transport of ions, which means that there's a random movement of these ions through the channels. There is no utilization of energy, which means that no ATP is utilized for the passage of these ions through the channels. And ions are transported across the electrochemical gradient or due to the potential difference across the cell membrane. And this concept of the electrochemical gradient or potential difference is explained in the video of simple and facilitated diffusion. So if you guys want, you can check that video. Classification of ion channels include classification on the basis of number of gates, on the basis of localization or location of proteins, on the basis of type of ions that are passed through the channels, and on the basis of gating. Whereas channels on the basis of type of gating are of two types: voltage gated channels and the ligand gating channels. Gates are the gate-like extensions of the transport proteins which can close the opening of channel or can be lifted away from opening by conformational change in shape of protein itself. Okay, this diagram shows ion channels, the transmembrane or the transport proteins, whereas this part is that gate-like extension which is called gate. And these gate-like extensions, they can close the opening of the ion channel like this as shown in this diagram, whereas by conformational changes in this protein, this gate-like extension, it can be lifted away, and that lifting away of this extension, gate-like extension, will result in opening of this channel and resulting in the passage of ions across the ion channels. Okay, so now we know that gates are the gate-like extension of the transport proteins, whereas gating of the protein channel is the process of opening and closing of the gates, providing a means of controlling the ion permeability of channels. So these are the gate-like extensions, whereas the process of opening and closing of these gates is called gating. Okay, membrane channels respond to chemical or physical changes in the environment around that particular channel, which then stimulate the opening or closing of channels. Whereas this opening and closing of gates are controlled in two principal ways. Number one is the voltage gating, and second is the ligand gating. Okay, voltage gating is the opening or closing of gates due to change in the molecular structure or chemical bonds as a response or as a result of the electric potential difference across the gates or across the ion channel. For example, we have sodium channels. This diagram shows the sodium channel in resting state. In resting state, we have more negative charges on the inside of the membrane and more positive charges on the outside of the membrane. As a result of these charges, or in resting state, the protein channel is closed. This channel is closed, due to which there will be no passage of the sodium ions. But on arrival of stimulus, what happens, that depolarization occurs. A potential difference is created across the membrane, and we get more positive charges on the inside and more negative charges on the outside of the cell membrane. And we know that with it's a very basic concept in physics that when we have charges, there will be an electric field associated with it, and that electric field runs from positive towards the negative. So when an electric field, as a result of this depolarization, the associated electric field here will induce conformational changes in this protein, and that conformational change will result in the lifting away of these gate-like extensions, resulting in opening of the channel, and that will allow then the passage of sodium ions from outside towards the inside of the cell membrane. Ligand gating is the opening or closing of the channel gates due to binding of a chemical substance called ligand with protein, causing change in conformation or the chemical bonds of protein channel. This diagram here shows ligand gated channels. This is that transmembrane protein which is acting as a channel. This part is that receptor site to which the ligands bind. These pink ones, the triangle shaped, they are those ligands or chemical substances that bind to the receptor site of the protein channel, and the blue ones are those ions that will be passing through these channels across the cell membrane. So before binding of this ligand or this chemical substances, the channel is closed, but as a result of binding of these ligand with the receptor site, as shown in this diagram, ligand binded to the receptor site, what happens that conformational changes occur. The shape of these protein channels changes, resulting in the opening of this channel and allowing the passage of ions or molecules that are to be transported across the channel, through this channel across the cell membrane. So this was all about this topic. Please don't forget to like and share this video and subscribe my YouTube channel. Thank you, take care, and Khuda Hafiz.